and also and the collection of drug response information to learn the relationship here, and then apply the relationship matrix to the patient clinical genomic or molecular profiles, which enable us to impute potential drug response in patient. So you can see we are applying this to several different areas of interest. For one, well, again, if we build a relationship in these large collection of cancer cell lines, learn the relationship between molecular profiles and drug response, we can then impute drug sensitivity in patients. So for every single patient-directed tumor, we can project over several hundreds of different drug sensitivity in that patient, as long as we know the molecular profile of the patient. Once that's done with the imputed drug sensitivity, in case of TCGA, for example, we have all the genomic information, for example, copy number variation or mutation. We can then do biomarker discovery. In this case, I'll show you the first application. So first thing we did is we actually call it a imputed drug sensitivity wide association studies. So similar to GWAS, genome-wide association study, here the phenotype, we swap it out by these imputed drug sensitivity. And we wanted to know if we can impute drug sensitivity in a piece of tumor. When we do association study with different molecular marker, can we find something that's already known in the clinic? So this work is mostly developed by one of the previous postdoc, Dr. Paul Gillard here. And we're looking for first proof of concept. This is one of those examples. Nutlin-3 is a drug that is a p53 inhibitor. So in this case, if we imputed the Nutlin-3A sensitivity in TCGA tumors, again, the score just want to oriented you, the higher the score, meaning more resistant to this medication, the lower the score will suggest more sensitive towards the medication. In this case, if we stratify patient based on their p53 mutation status, you can see there's a higher average score in the mutated tumors when compared to wild type p53, and this is very significant. And the results suggest p53 mutation is a good biomarker in the sense that if you carry wild type, you're more likely to be sensitive to this medication. This is a proof of concept. And the other view to see this is a frequency distribution. Basically, we're asking the question, the mutation of every single gene in the genome in association with the imputed Nutlin-3A sensitivity. You can see p53 is one of the most significant biomarker that's associated with this particular drug. For most of the gene we interrogated, they do not show their genetic variation or mutation in the gene does not show association to Nutlin-3A. And Nutlin is not the only example. We have a number of other drugs. Again, the same format. For example, here, bemarituzumab is a well-known BRAF inhibitor, and you can see BRAF as expected. The mutation in BRAF is significantly associated with imputed bemarituzumab sensitivity in these TCGA cancers. Not only of course, it's good to have the proof of concept, but we're more interested in discovering new biomarkers. So this is one of the example of work we've done associated with both the discovery and validation of the biomarkers. In this case, we're focusing on a drug called veneralbin. So this is a vancomycin. It's a microtubule inhibitor which has been used to treat breast cancer. In this case, we found out a copy number variation, in this case, amplification of a ERLIN2 gene. The increase in copy number is associated with higher predicted scores, which suggest more resistant to this particular drug, veneralbin. At the time when surveying literature, there's no suggestion about this particular gene copy number variation. It's associated with the drug. However, mechanistically, the particular gene has been suggested to play a role to stabilize microtubule. And in this case, the drug is target these stabilized microtubules. So it makes a lot of sense that we see this association. So in the laboratory, we actually use a breast cancer cell lines, and we overexpress ERLIN2 gene. This is in the DNA level, and panel B is on the RNA level, showing there's a clear overexpression of ERLIN2 gene. And when you take a look at the panel C, so all the dashed line are basically these breast cancer cells without the drug exposure. You see introducing additional copy of ERLIN2 gene that literally did not change things much. However, when you pay attention to the solid line, all these solid lines are exposed to the drug, and the green and the black lines are the control, and the red line being overexpression of the gene of interest, ERLIN2. You can see there's a clearly increase in resistance to vinorelbin in the presence of overexpression of ERLIN2 gene. So this is a experimental validation of the biomarker we discovered. To summarize this part, we can convert TCGA or many other clinical cancer data sets alike into large pharmacogenomic data sets. We have confirmed some previous known gene-drug relationship. We also discovered many new biomarkers. This is one of the ways to, nowadays we, as the field, we care a lot about finding the proper biomarker, having the companion diagnosis kit to make sure our drugs are given to the right patients. I think this presents a unique opportunity because for many of our biomarker discovery work in regular clinical trials, your sample size usually are in the hundreds, a few hundreds. In this case, we're significantly increased the sample size. For example, TCGA, we're dealing with over 10,000 samples that really enable us to much higher power to discover pharmacogenomic biomarkers. So the second application I want to point out, by employing such approach, again, we learn the relationship in preclinical data sets, and then we impute the drug sensitivity in patients. But the next thing we care about is, well, if we can stratify patients based on either the subtype of disease or their response to standard of care medication, we can then find a drug that could potentially be efficacious in specific subtype or certain patient cohort that was resistant to standard of care treatment. In this case, we care a lot about drug repositioning. Why drug repurposing? We are all familiar with traditional drug development. It's a lengthy process. It takes over 12 years and more than $2 billion to bring one chemical from the preclinical discovery stage all the way to market, phase 3 and post approved drug approval. And more importantly, the failure rate for new drug development has been really high. Only less than 10% of the drug make it through the whole process. Drug repurposing, in a way, is to bring some of these, in our definition, we are trying to look for a drug that's either already FDA approved or in the late stage of development, already in clinical studies. We can see, so many of these drugs has already gone through the toxicity and other kind of assessment in humans. So the whole process could be significantly shorter. And in this case, the two preclinical high throughput drug screen done in Sanger and Broad has also screened many of the compounds that's already in the market. So this is an example, again, done by talented postdoc and the graduate student, Robert Gruner here. When I say, we imputed drug sensitivity, so every dot's representing a potential sensitivity to an agent in a collection of patients. In this case, breast cancer. So this is the volcano plot on the panel A. Basically, we ask the question, in breast cancer, triple negative breast cancer is one of the ones that's most problematic because for the hormone receptor positive disease, we as a society has some very efficient drug to treat them. But for TNBC, as of today, there's no receptor on the surface of the cancer cells. Therefore, most of the therapy are still limited to toxic chemotherapy. So we ask the question, if we compare receptor positive breast cancer to triple negative breast cancer, we're looking for drugs that's showing higher imputed sensitivity in TNBC when compared to the other subtypes. And you can see there are a number of drugs showing that significantly higher sensitivity. Again, the higher the value on the right, suggesting higher sensitivity in TNBC, and the Y is the p-value in comparison. So among those, the very top drug, the AZD7762, is one of the agents that indeed has been evaluated in the clinic. It actually showed efficacy, but the problem is the higher side effects. And I want to bring your eyes to this particular agent. Again, among the top 3 efficacious agents projected by our work. So we break breast cancer down into different subtypes. The panel B is based on a reported patient subtype, either ER/PR positive or triple positive, or just HER2 positive. And you can see TNBC showing the lowest imputed score, which again suggesting higher sensitivity. And this doesn't matter, we break it down into just based on receptor status or PAM50 status, always this particular agent showing higher efficacy in TNBC. And this approach, one of the strengths of this approach is that, in your regular drug development, you're seeing finding the target and then finding drugs that's target that specific target. In our case, we're finding drug that's hitting variety of the different mechanism. So this discovery is enabled not target driven, but rather the collective of transcriptome representing drug sensitivity. So then what we did is we take our initial discovery into validation process. We did the work both in vitro, in cell line setting, as well as in animal. So this is one of the TNBC cell line that over time you can see the cell growth. And in the presence of paclitaxel, which is the standard of care agent, you see initially the cell growth was inhibited, but over time that cell recovered and growth is catching up back. And this blue line is the drug of interest, the AZD1775. We see over time there's a gradual decrease in the cell proliferation. When we put the standard of care agents and our drug of interest together, the cell lines growth has been inhibited over time. And this is not only the case in cell line model, this is the experiment in xenograft. So basically we put TNBC cancer cells into mice, and the black line, so on the panel on the left, it's showing measuring tumor volume. You can see again, without any intervention, the tumor grows over time. And in the presence of standard of care paclitaxel, there's a small decrease in the growth, inhibit the growth of tumor. In the presence of our drug of interest, AZD, also there's a inhibition of growth over time. And when putting the standard of care and the AZD1775 together, you can see the cancer cell growth has been inhibited in these animal model. And when we look at animal survival as a phenotype, again, this showing that the combination therapy can significantly slow down the proliferation of tumor in the animal. So that going forward, we're doing work not only in breast cancer, we're also interested in metastatic castration-resistant prostate cancer, brain cancer, ovarian cancer, and some other rare cancers. The conclusion for my talk really is that the novel computational tools built using these preclinical drug screen data sets can enable drug sensitivity prediction in patient tumors. When we apply this to large clinical data set, we can generate new large pharmacogenomic data sets for biomarker discovery, and also use it for drug repurposing and drug discovery. We believe that these computational tools can enable the discovery of new drug with variety of different mechanisms. So with that, this is absolutely a teamwork. I want to acknowledge my lab and many people in the lab, as well as our many collaborators and the founding resource. Thank you very much. I guess I'll take your question. Feel free to type in the question in the chat. We'll discuss this in more details in the panel discussion site. Thank you.
Oh, thank you, Dr. Huang. This is a very nice presentation. So actually, we probably have a few minutes for questions. So if you have questions, yes. So there is a question in your talk about drug repurposing. What data source did you need?
Yeah, so actually, I didn't go into much of the details, but when we build a model, we in preclinical model, we have tried using DNA level information. We have tried it using RNA level information. So in our hand, the RNA information can capture majority of the drug sensitivity variation. So our model really, even though I put the molecular profile I'm referring to is transcriptome data. So we need gene expression information, either from preclinical model or from tumor. At the time when we first developed this model, transcriptome data are not always there. In cancer nowadays, a lot of the genomic information are there, but with the time progression, we've seen more and more of the transcriptome data that's available. We have, since our initial launch of the method, we have applied this to many other clinical data set that has transcriptome data. So on the clinical side, all we need is the transcriptome data. For the development of models for preclinical, we've tried a variety of different things. Again, I think transcriptome data is work out the best. And regarding drug sensitivity, we have used your standard either IC50 or area under the curve, which basically, you measure cell growth inhibition with increasing concentration of a drug. So IC50, that's the concentration inhibit 50% of the cell growth. AUC is usually referred to that area under the drug response curve. So bigger the AUC, the more resistant that compound is to the cell. Yeah.
Cool, thank you.
Yep.
Another question, are there drugs approved using repositioning approach? I think this is a very important question for all the drug repositioning type of research.
That's right. I am not aware of right away approval, but the good thing is, the more we do this, I'm really confident that, the things that we found really show, so far, I think in a way we're lucky, we're cherry picking on the top signal. So most of the drug that we chose to move forward was experimental validation. So at least for my lab, most of the work are right now in the animal stage. So when we discover the things, we move it into usually your preclinical screen and then into animal. So far, we're lucky in a way that all the animal experiments have cell line experiment and animal experiment has panned out and showing efficacy as we predicted using these computational methods. The other thing I really want to highlight, I truly believe this could shorten the time of discovery because, you think about your regular drug development, you want to find out what differ between the disease and the normal. So finding that target and then you screen thousands of compounds towards that single target or few. We know cancer is such a complex thing, so it's not going to be one target. Even with the FDA approved drug, they're targeting something, but we all know there's multiple other off-target effect or maybe potential true hidden mechanism that leads to efficacy. So in our line of work, we don't really say specifically one gene is important. Every single gene in the transcriptome were taken into the model. So we just let the data speak which one played higher weight or more important in these drug sensitivity. And that's partly I see our final candidate drug lists are usually not just hitting one pathway. It's has a good representation of variety of different things.
I see, thank you. And probably we have time for another question here. And so the question is, have you considered non-clinical data like social factors or demographic characteristics of the patients? And how would you think about this approach being applied in the community setting?
Yeah, so it's a very good question, and that's very practical. I can see the question asked person has translation in mind. At the time, we did look, the patient demographic information. Those are the things that we have those tools with, for a long time, we have tried to utilize. I think in my mind, these factors are important, but they're not enough to accurately predict a drug response. They explain part of it, but not at a finer resolution to pinpoint each individual. And honestly, the nice thing about transcriptome is transcriptome can capture a lot of these information. So because we're using transcriptome, we can actually see that capture what tissue origin, the expression of a tumor would be a representing of many of the factors that we're interested. So in our hand, we have looked at regular clinical traits. We have looked at using DNA genomic as a predictor, using transcriptome as a predictor. In this case, transcriptome works out the best in finer resolution in projecting a patient response to medication. I'm hoping down the road with all these molecular technique become cheaper and cheaper. Honestly, with DNA information 10 years ago, we think that's such a fancy thing, but nowadays it can be done so easily. So I'm hopeful that transcriptome or other things could be measured in the clinic down the road as well.
Thank you, Dr. Huang. So thank you for the very nice presentation. I believe there are more questions during the panel discussion. So yes, probably you have 2 minutes for one more question. Are you muted?
So I'm thinking, for talking about targets, right? But usually we can identify, genetic mutation, usually we can narrow down to a few, right? But in terms of methylation or differentially expressed genes, we always can easily get a large number, hundreds, right? Targets. So do you have any ideas, say, if you mention, say, if likely to pick up some cherry picking to get some good results, do you have any advice? Say, you also have this kind of challenge. How can you pick up the right ones?
Yeah, I think it's an excellent question. Honestly, the biomarker discovery approach we propose, we can find things for sure, but it's also tinkling my mind. Again, cancer is a complex disease. We're doing this one gene at a time for association. So I'm pretty confident the things that we found to significantly associate are real, but where do you cut that line? How far do you go down the list? And I truly believe it's not gonna be, even though right now, when we approve a drug, it's still one gene or whatever, one biomarker was the drug. I think that down the road, it's going to be a multi-polygenic sort of risk thing. At least that makes sense to me. It's not going to be a single gene or just one layer of mutation or something.
Thank you.
Thank you. Perfect. So we're right on time. Thank you again, Dr. Huang, for the nice presentation. And we'll move on to the next one. And I believe there are more questions for you during the panel discussion. Thank you. And the next presentation is from Dr. Li. So Dr. Li is assistant professor at the Institute for Informatics and Department of Pediatrics at Washington University School of Medicine in St. Louis. His recent research has focused on the integration and interpretation of diverse and heterogeneous pharmacogenomics data for understanding driver genetics, functional mechanisms, and discovering novel combinatory therapies to overcome intrinsic and acquired drug resistance, and also uncovering and perturbing the tumor stroma interactions to understand the rules of tumor microenvironment in the disease development. So Dr. Li, you have 30 minutes.
Thank you, Dr. Xia, for the introduction and the invitation, Dr. Li. So I'm glad to also share some of my work to especially to predict the drug combination because as Dr. Huang said, cancer is really have complex signal pathways dysfunction. So probably single target, or it's hard to first. I think it's hard to pick up the right target, and second is it's hard to inhibit just one target to have good drug response. Yeah. So and also in this presentation, I want to say focus on more about interpretable network analysis for drug combination prediction. So why drug combination prediction is important? There are quite a few reasons, but I think the most important ones are the intrinsic or acquired drug resistance are still one of the major challenges in treatment of complex disease. Not just like cancer, but also many diseases involves a lot of complicated signal pathways. So how to overcome or delay drug resistance to enhance treatment response, definitely I think drug combination could be one possibility. So the other is, why prediction is important, because there are a large number of possible drug combinations from, there are about 3,000 FDA approved drugs, and also there's over much more, we call it investigational agents. That means these compounds or agents have been well studied. For example, they have been tested in toxicity studies, experiments, and they are safe, but they are not finally FDA approved because they may fail in clinical trials or other different considerations. And so it's limited to experimentally screen effective drug combinations because of first, whether there's good model, like a preclinical model or not, right, to mimic the disease perfectly. For single drug, you can screen 1,000 drugs. I think it's doable, but for drug combination, it's hard to screen drug combinations in a unbiased way because there will be a large number of drug combinations. And so the prediction purpose is try to prioritize the possible drug combinations, and then you collaborate with experimental or biologists or doctors to validate your predictions. And why drug combination? Why is drug combination prediction challenging? Again, so this is my understanding is first, the mechanism of synergy of drug combinations is heterogeneous and hard to predict, and there's no well-known pattern, say mechanism synergy pattern to follow. Say, okay, this is the best way to identify drug combination for cancer A, for cancer B. It's hard because right now, because I think the first thing is our understanding of drug combination is very limited, and also the existing drug combinations, effective drug combinations in clinical side is still very limited. And the second is, again, cancer is really have complex signal pathways dysfunctional. I think this is why GWAS is really important and popular in clinical side, because doctors or clinical faculties believe, okay, this is if a patient have, like for melanoma, if there's a BRAF mutation, yes, this is one subtype of breast cancer. There's ER, HER2, or triple negative of breast cancer. And with this, a few biomarkers, people can understand it, and they can say, okay, this is the causal genes causing drug resistance or different drug response. However, the reality is many cancer patients, even they bear some genetic mutations, and then drug sometimes doesn't work, or the second is there's drug resistance. And for many other cancer patients, there is no specific druggable genetic mutations. If we come to, say, integrative analysis to combine genetic mutations with gene expression, methylation, proteomics data, copy number variation data, microRNA data, it will make the model is really hard to interpret. And because we cannot understand, say, how hundreds of genes work together to cause the drug response. No one believe it. No one can understand it. So This is why it's challenging. The second is because again, so there will be millions of possible drug combinations, even for pairwise drug combinations. So the challenge is it's hard to predict only a few, right? You cannot tell your collaborator, say, I rank the top, 5,000 drug combinations you can test. No, they want to say, hey, can you give me 5, right? And from millions to pick up 5, statistically, you have very high possibility to be wrong. So I think this is also very hard. So I think this is one paper in 2009. I think when I read this paper, I felt that's very interesting. In this paper, they tried to investigate all the existed or reported synergistic effective drug combinations, try to understand how's the possible. They try to find, say, whether there's a common pattern in terms of mechanism of synergy. And they trust to summarize it into different categories. For example, the drug combinations, target on the same signaling pathways or different signaling pathways, the same target on the same pathways or different targets on different signaling pathways. In another way, the conclusion is it's really hard to get a conclusion, say which whether there's common mechanism of synergy. So in other words, so we can get nothing. Although we thought we can get something, but actually it's really hard to get something. So ideally, so it tells us ideally effective drug combinations is can say inhibiting multiple essential targets on the essential signaling pathways, but the challenge is the same as precision medicine. The precision medicine is defined to give the right drug to the right patient at the right time. But we don't know what's the right drug, what's the right time, and what's the right time, and what's the right drug. So similar as here, drug combination is here. We don't know what's the essential targets, and we don't know what's the essential signaling pathways. It's intractable. However, say computationally, we still can do something. So basically we can say, okay, heuristically, we can do tractable solutions by identify drug combinations inhibiting multiple signal pathway or signal network modules. And this is the basic idea. In 2014, so we published the first paper based on this idea. So basically we try to say we cannot like Dr. Huang's work, say identify a couple of biomarkers, right? But we can get a single network using single pathway analysis, goal term analysis, or differential expression analysis. And we want to classify or cluster these set of disease genes into different network modules. And the basic idea is whether we can identify these disease-associated network modules with different drugs. So in another word, so we try to pick up different drugs have with different mechanism of synergy, a mechanism of action targeting on different parts of network. Then they probably have higher possibility to be synergistic and effective. And it turns out our prediction models can work kind of really good on different cancer subtypes, of course, but we don't have the experimental validation in this work because we are pure computational side. And then on the another work, so we want to identify effective drug combinations for melanoma because the drug resistance, acquired drug resistance will occur in probably a few months for melanoma treatment. And the basic idea is similar. So first, we want to identify the dysfunctional single network. On the other hand, we also want to cluster drugs into different clusters because we easily can get, still can get 100 drugs could be related to this single pathway, so single network. Then we cluster these drugs based on their similarity of their drug response profiles from like a CMAP, connective to MAP. And then we got, for example, 19 drug clusters. And then for drug combination, we pick up drugs from each cluster to test what's the best drug combination that could be able to inhibit different parts of the dysfunction network. And then we got a set of drug combinations top ranked. So one top-ranked drug combination is vemurafine and tretinoin. So these two drug combinations, were number one ranked in our prediction, and through experimental validation, we found that this drug combination can really inhibit the drug, the cell line model A75 cell line BRAF melanoma cancer cell line. So, and then we want to know in another work, so we want to understand, say, similar. So how can we identify the single network, right? Modules. Yes, there are some, you can depart the network into different modules. But on the other hand, we are thinking, see, can we just based on the prediction, top ranked drugs, divide these drugs into different drug clusters? For example, in this work, just based on that the target, because for many drugs, for example, CMAP is a good data resource, but for many investigational agents or FDA products, they don't have this genetic response profiles available. So we want to see whether we can just use drug target, right, to cluster these drugs into different modules. And then we pick up, drug combinations from, individual drugs from different drug clusters, and then see whether drug combinations from with drugs from different drug clusters, and that these drugs also can inhibit this disease signal network. So that means first, we can divide the signal network into different network modules. But on the other hand, we can also divide the drugs into different drug clusters. So we found that interestingly, in the top-ranked drug combinations and validated drug combinations, many of these effective, experimentally in another independent work, validated drug combinations, most of these effective drug combinations are from different drug clusters. This is all these few examples, we want to see is these examples are heuristic. So based on what we believe and what we observed, the basic idea is we try to identify drugs that target on different parts of the single network of disease. And then recently, definitely predictive models, especially the artificial intelligence model or deep learning models become very popular over that can have much better results compared to existing machine learning models. So we want to ask the question is, can we train deep learning or AI models to predict drug combinations? I believe, most of you will believe, yes, it definitely can do the drug combination prediction. The second is whether it can learn the mechanism of synergy. And that's very interesting for us. So because we want to understand, what's the real targets that play important roles causing the regulating drug combination response. So of course, if you want to train the model, you have to use some existing label data to guide your model, to train your model. So this is one example. So there are a few data sets available in terms of experimental drug combination screening data sets. So this is one interesting data set, NCI, MANIC. So for this data set, it's generated in 2017, and it have the pairwise drug combination screening data of over 100 FDA-approved drugs. And drug combinations were evaluated on 60 cancer cell lines. This is NCI 60 cancer lines, including multiple cancer subtypes. So this is a very good resource to train a model, to test the model. So basically for the, in terms of the data, you can think about it as using 4-element tuple to represent this drug combination screening data set. It's drug A, drug B on cell line C with a synergistic score on ABC. So this data set is publicly available. And to answer the first question, whether deep learning or AI model can perform better in terms of drug combination prediction. So, and the deep synergy is almost the first paper that published to say we use a very generic or very standard deep learning model. So basically, they integrate the chemical structures of two drugs or two compounds with the genomics data of an individual cell line. So basically, you will have over 10,000 features to predict a drug combination response on a specific cell line given two drugs, drug A and drug B on cell line C. So and then this model can really outperform other existing models in terms of drug combination prediction. So we also developed a deep learning model. In addition to using chemical structures of drugs and also genomics data of cell line, so we also integrate like the TCGA data. We want to identify, all the important, we call it a kind transfer lineage, because we want to train the autoencoders using TCGA data, copy number, expression data, and mutation data. And then for the new cell lines, we will convert the input of new genomics data of copy number, mutation, and gene expression data through these autoencoders. And based on that, these autoencoders were trained based on TCGA data. So why not we can directly concatenate all the genomic features of cell lines as the same as using the same structure as deep synergy, because this is a challenge. In deep synergy, they use all the gene expression data. For example, there's about 20,000 genes expression data. But if you also consider copy number, genetic mutation, and also the gene expression data, there will be 3 times larger the input. So the number of parameters will be huge. So this is what we don't want to do, because there are too many genetic features, and there are only a few available drug combination training label data to train a model. So that means the trained model will be sub-optimized. So it's hard to see whether your model really can get the point to predict the drug combination. And then, so we want to do better. The second question, can we learn the mechanism of synergy? So this is the 2 papers in 2019, very nice papers, because what? Because it's very neat and clean. So basically, they want to see why drugs that inhibiting RAS pathways doesn't work for many cancers. Because RAS is really one of the most important genetic mutations or dysfunctional biomarkers in many cancers. But we have, at the same time, we have drugs, compounds, investigational agents that can be able to inhibit RAS signaling pathway. However, many of these drug combinations, many of these drugs, single drug inhibiting RAS doesn't work at all. We don't know why. Suppose, this is the causal gene, right? We inhibited this causal gene, the tumor still is there and keep growing. We don't know why. And then in these 2 papers, they found that, okay, once the RAS signal pathway inhibited, autophagy signal pathway will be activated. So that means you have to inhibit both autophagy and RAS signal pathway. Then you can solve the problem. It's very simple. I mean, the mechanism is very simple. The pathways involved is very simple. But the question is, how can you identify these 2 pathways and the 2 targets? And so in another paper in 2018, this is very interesting. They studied the cancer, all the cancer samples in TCGA, and they found that using only 10 single pathways, it can cover that 89% of tumors have at least 1 driver alteration in one of the 10 single pathways. And 57 of tumor patients have at least 1 druggable target. And 30% of tumors have multiple drug biomarkers could be targeted by drug combinations. So that means what? That means the single pathways are usually not so complicated. We only have like 10 single pathways. But how can you identify the right targets is important. So that means the single pathways, single cascades are very complex, but single networks probably is not so complex. So we want to see, can we learn this single mechanism synergy to identify drug combinations synergistically? And so this is two papers, very interesting, D-cell and drug cell, published by Tree Edicus lab. So basically they propose a new network concept. I mean, probably it's not new, but at least interpretable. They call it a visible neural network. So basically, in terms of using black box of AI models, they try to design the network model based on biological meaningful things. For example, gene A connects to gene B in the network, but also gene A and gene B belong to Go terms. So they try to have a hierarchical, the key is hierarchical network structure. It's defined, it's predefined rather than learned by the network. So then they try to learn this network, to train this network using, for example, survival analysis, drug response data, try to identify the most important targets. Okay, then it can be interpretable to predict the drug combinations. And we also did some work. So we found that we believe that, okay, this network is still very complex. And also we believe the pathway is the most important part rather than, you know, other general relationships. So we only want to focus on pathway. The first work we worked on is we want to see in terms of using all the genomics data, whether we can only use 46 pathways reported in CAG pathway database. So this 46 pathway related to cancer a lot. And for each gene, easily you can integrate the gene expression data, that whether it is the drug target data, whether there's a genetic mutation or methylation, or all the genes features can be integrated naturally into this model. And based on this very small set of genes, we can also achieve very good drug combination prediction. And also this network is still not well interpretable because although we did the interpretable analysis, we found that a lot of single pathways, rather than the key single pathways, other single pathways play important roles in drug combination prediction. However, it's still a challenge is many genes, for example, KRAS genes may be involved in multiple single pathways. It's hard to say it's because their single pathway is important or because their target is important. So we improved our model again using pure network analysis. So, and the good thing is you can say, so single pathways naturally itself is a graph, right? Of course, you can use graph network. And the second is the activity status of individual genes is decided by multi-omics data. So you can easily use the node features like gene expression, common number variation, genetic mutation, and also even microRNA. You can integrate these multi-omics data as the features of each individual gene. We call it weighted bidirectional GCN. Because usually the general GCN just try to, the same as like a convolutional network or image, it's just combine the neighbor information evenly to each individual node. But we think it should not be even, it should be weighted because, for example, many genes linked to many other genes, but the signal transduction, signal cascading on this network should be unevenly. So for example, some gene interaction is important in some cancer subtype, but some are not. So we add a weight here. And why is bidirectional? Because two reasons. Because you inhibit a target, there are two parts. One is upstream. Upstream is signaling transduct to the drug targets, and there's downstream from drug targets to the downstream. So we call it a bidirectional. We build two network models. One is upstream, the other is downstream, and integrate multi-omics data. And this is the basic structure. We can go to the results part. Of course, you can get all the gene features from a lot of CCLE, a lot of database drug combinations, drug target, and also single pathways. The reason right now we're using CAG because two reasons. First, it's directional. The second is has a small scale. Because we are testing on the general protein interaction network, but it takes a long time to train the model. So these results we can say. So this we can see the prediction first. It has the capability to predict drug combination at a very good accuracy compared to deep synergy. without considering. The most important thing is through this model, we can identify the potential network. So first, we can predict which interaction is important. So from this part, two images, you can see this is upstream and this is downstream. And upstream means from other genes to the drug target, and the downstream from drug target to other stream. So the width of the interaction edge is indicating whether this interaction is important or not. So the targets selected from 1,000 or 2,000 targets, we think this is the most important targets, and their interactions are important. So we combine together, so we can say we combine the upstream and downstream together, we can conclude that for example, for some specific cancer subtypes, this is the key mechanism of synergy to cause the drug resistance or drug combination resistance or the synergy of the drug combinations. So right now we are trying to make a further analysis to see whether we really can give examples why some specific drug combination really works better than others. Yeah. So the conclusion is the deep learning right now and AI models could predict synergistic drug combinations by integrating multi-omics data, right? Because one gene is easily can integrate multi-omics data and also single pathway information to learn the key targets that are interacting with each other to regulate drug combination effects. And also, we still can see there are a lot of challenges how to identify the really complex single pathway interactions on a very simple single pathways. We want to say whether we can use the model to learn that because of target A on pathway A causing the activation of target B on pathway B, and then we use the drug combination to inhibit both of targets can get a better drug combination response or synergistic effects. So this is what we want to do using AI to automatically learn these mechanisms. Yeah, but it's still quite hard. Yeah. Thank you. So this work, is conducted by two students, Heming and Jerry, and other collaborators, Kelly, Yixin, and Philip, and thanks funding from NRA and also Children Discovery Institute. Yeah, and again, thanks, Dr. Xia and Dr. Li for the invitation. Yeah, thank you.
Oh, thank you, Dr. Li. This is a very interesting presentation, and we do have some questions, but probably we'll defer all the questions in the panel discussion. So basically, it's about the number of parameters in your deep learning model, and
yes,
also some questions related to the the target combinations. So as for now, let's move on to the last presentation of this session. And thank you again, Dr. Li. So the next presentation is from Dr. Chen, and Dr. Chen is a principal investigator with the Cleveland Clinic's Genomic Medicine Institute. So Dr. Chen's research is focused combining tools from network medicine, artificial intelligence, genomics, bioinformatics, computational biology, chemical biology, and experimental pharmacology and system biology assays to address the challenging questions of understanding of various human complex disease. And for example, Alzheimer's disease, which could have a major impact in identifying novel real-world data-driven diagnostic biomarkers and therapeutic approaches for precision medicine. So there are several focused areas of research in Dr. Chen's lab, like network medicine tools for precision medicine drug discovery, translation of multi-omics discoveries to disease biology and therapeutic development, artificial intelligence methodologies for target identification and therapeutic discovery, and building an individualized network medicine infrastructure for precision cardiology. So Dr. Chen, it's all yours. Hello, Dr. Chen. Hello, Dr. Chen. My network or?
It was muted.
No, I think the
Can you hear me now?
frozen.
Yeah,
Yes,
yeah.
I can hear you. But hi, Dr. Chen. You are muted.
I think I not muted right now.
Okay.
Okay, go ahead.
I'm sorry, my laptop just frozen, just started frozen, but I just switched to my desktop. Hopefully you can see that. So thank you so much, Xiao, for great introduction and your kind of invitation to present in this fantastic meeting. So today I will share our recent work, how we use the network medicine and AI, methodology for drug repositioning. Just use the COVID-19 as an example. I'm Fei-Chun Chen and came from the Cleveland Clinical. So I know any computer interest need to disclose. So before my presentation, I just give very brief introduction about our Cleveland Clinical healthcare system. So our Cleveland Clinical is our largest healthcare, international healthcare system around the world. So our main campus located in the Ohio, Cleveland area, but we also have other hospital or in Florida or other state. But we also opened some a hospital in other countries, including London, just opened in this year, and Canada also opened in near future in Shanghai, China as well. So in the past few years, especially for these years, our Cleveland Clinic, we have multiple, large scale, AI and healthcare, data initiative. For example, just in the beginning of this year, we partnered with the, Ohio governor. We have like 500 million partnership, with the Ohio, developing novel technology, also include AI and digital health technology, help us to respond to future, the pandemic if we have, and we just, a couple of months ago, we also have, the big, partnership in the next 10 years with IBM, use the outstanding, AI, also include the quantum, computing platform from the IBM to analyze large-scale million or 10 millions, healthcare data will accumulate at our Cleveland Clinical in the next 10 years. For example, in the last year, I think in the early of March, we see first patient at our Cleveland Clinical at Ohio. Our Cleveland Clinical, we are rapidly moved to the COVID-19 research against this pandemic. For example, in the past years or maybe over than 1 year, we built a larger scale cohort, include the acute COVID-19 cohort, also post-acute COVID-19 cohort, use the digital healthcare data. We not just work on the clinical, the clinical informatics, study. We also generate larger scale omics data from the biospecimen, we collected from the COVID-19 patient as well. So let me come back to my lab. So my lab at Cleveland Clinical, we use the system biology and network medicine technology to predict drug target and understand the mechanism for the human disease. For example, we work on, we call the cardiac oncology, means we try to predict cardiotoxicity for cancer patients during the cancer treatment. We also work on pharmacogenomic study, especially work on cancer pharmacogenomic study. But today I'm going to share our recent story, how we use the network medicine and AI methodology for drug repositioning or drug combination design. Just use the COVID-19 as an example. So this slide just shows the methodology we concept, presented by, fantastic, talk by Dr. Huang and Dr. Li as well. So the idea we use is we take in the multi-omics data, include the DNA level, RNA level, and the protein level, generate from the patient biospecimen, include, cell, tissue, organ, or organism. And then we apply the different network-based approach or AI, include the deep learning approach to identify the novel target or predict the drug candidate, including the drug repositioning for disease treatment. And then we use the different, cell model, animal model, also include the larger scale, we call electronic health record data to validate our prediction. If we get successful, we're making rapidly move to the clinical trial testing. Today, I mainly share our, we apply this kind of idea for the different disease area. For example, we work on the neurodegenerative disease like Alzheimer's disease. But today, I mainly share our, recent story, how we respond to the, COVID-19. Just use the COVID example. So idea we highlight here is, we argue, genetic or genomic, of course, is very important for human disease, study included the, precision medicine. But we know that also many other, environment factor or exposure are also important for complex disease as well. So our idea is not just study the genetic or genomic, our hypothesis is the interaction between genetic and genomic may generate a unique what we call human cellular network or human interactome. So we try to use the human interactome network to help us to better understand the mechanism of human disease or identify the drug target for human disease. So this is the network medicine approach, we just highlight here, so our lab is we build a comprehensive we call physical protein-protein interaction network. In this video, each dot, just the protein and the connection, just shows a physical protein-protein interaction. So our lab is trying to develop a new algorithm, network-based algorithm, try to integrate what we call multimodal omics data, include genomics data, transcriptome data, also proteomics data, to this comprehensive human interactome network to identify disease module we highlight by the the pink module here. Use this pink module to predict a drug target or identify the potential mechanism pathway in human disease, not just focus on the single mutant gene or single variation. In the next 20 minutes, I just give 3 examples how we use the network-based approach, AI-based approach, or drug combination design, how we can use this kind of idea for COVID-19 just as an example. So my lab work on the drug repositioning or drug repositioning study almost more than 1 decade. We apply this idea for multiple disease area. But today, and right now we are mainly focused on neurodegenerative disease, and we just quickly or rapidly move to the COVID-19 use the similar idea we used before. So for COVID-19 study, our idea is not just work on the antivirals drug targeted therapeutics. So we are more focused on we call host targeting therapeutics. We try to targeting the host factor that we call human protein or gene, this host factor, and then we may potentially treat the COVID-19 as well. For this kind of idea, first we have to build what we call comprehensive SARS-CoV-2 interactome network. The left two, the interactome just built by Dr. Krogan from the USSF, built the comprehensive what we call or variant protein interactome. And the right just our ongoing project with Dr. Yu from Cornell. We also built our own we call binary SARS-CoV-2 variants, host interactome, use the different protein-protein interaction assay. So my lab is we trying to integrate or leverage different type of omics data, include the SARS-CoV-2 variants host interactome I introduced in the previous slide. We also integrate different transcriptome or proteomics data generated from COVID-19 or SARS-CoV-2 positive patient compared to the control or across the different disease progression. Integrate all this kind of interactome or omics data into the human interactome network I introduced before to identify the novel host target or identify potential therapeutics or repurpose drug, and then we use the COVID-19 EHR data or registry database for validation. So for example, we learned a lot for COVID-19 in the past year is COVID-19 is really complex disease. It's not just the pulmonary disease or the infection disease. They also cause many other different comorbidities, including heart problem, neurology problem, also autoimmune disease as well. So for example, last year, two the fantastic postdoc in my lab, Yadi and the Yuan, they use the the network proximity approach we established to systematic way to look at how the SARS-CoV-2 variant host interactome overlapped with the different disease module we identified. What we found is SARS-CoV-2 variant host interactome are highly close or overlapped with the different disease like cancer, cardiovascular, also include the neurology disease as well, we just published. So this kind of idea help us to understand why the SARS-CoV-2 are highly overlapped with other disease. And for other disease, we know other diseases have FDA-approved drug available. So this come out of our concept is, the FDA-approved drug for other disease may, potentially treat the COVID-19 as well, because COVID-19 or SARS-CoV-2, the infection, they share the similar or overlap the pathway with other, human disease. So why we can do that? Because, like Dr. Huang mentioned also, some, fantastic work is, FDA-approved drug not just target the single protein, they target the multiple protein in our human body. For example, this, the drug targeting network, when I, PD training, we build, for example, the square just to show human, protein, and the circle just to show FDA-approved drug are grouped by, different ATC code. For example, this drug, cancer drug, this is the cardiovascular drug. So what I really learned from this drug target network analysis is FDA-approved drug not just target a single protein, they target the multiple protein in our human body or our human cell. So combine all this kind of together, my lab, mainly work on what we call the network medicine research field. So network medicine is trying to redefine human disease and therapeutics using the system biology, AI, and network, science technology, try to identify testable or actionable drug target or therapeutic approach for human, disease, including the COVID-19 as well. So our idea is for the complex human interaction network, we can identify the disease module. And for different drug, we know the drug target in human cell. So we can look at this particular drug target network distance with the human, with the disease module in the human interaction network. For example, COVID-19, as I mentioned before, we test one idea we call host targeting therapeutics by targeting SARS-CoV-2 interaction. So our idea is we try to identify FDA-approved drug. This drug can, block or inhibit the host factor or the variant-host interaction for coronavirus and a human gene and a protein. So the approach we use, like I mentioned before, we call the network proximity approach. We identify the COVID-19 module from the SARS-CoV-2 interaction or from the transcriptome data or proteomics data from the SARS-CoV-2 positive patient compared to the case control. And then we look at the different drug target network distance with the disease module. So this is very similar concept like the GPS when you use the transportation network. When you drive the car, you can use the GPS to help you to identify the short distance path between your study, to your destination. So for example, here just to show the network proximity predicted results, we just highlight 16 top predicted drug. This drug can target the SARS-CoV-2 or other human coronavirus and as well use the network proximity approach. And the different drug we just grouped by the different category. This is similar idea we showed before. You can see the different type or diverse pathway drug may potentially treat the COVID-19 as well, because COVID-19 is a really, really complex disease. They have impact in multiple human tissue or organs, we showed before. So the question coming to us is how we can validate this result or how we can validate network-based prediction. So we try to leverage, I mentioned before, use the largest scale electronic health record data we generated by working with Dr. Laura Jeha is our chief informatics officer. So we build a large scale COVID-19 registry data. For example, right now we have totally more than 70,000 COVID-19 positive acute and post acute COVID-19 patient sample available. So how we do this? So we try to use, for each data study, we know the limitation, especially for confounding factor or other missing data issue. So we try to use the probability score calibration approach to design the different drug cohort. For example, we focus on the targeted drug like melatonin is a very top predicted drug from our SARS-CoV-2 interaction-based prediction. We also compare the melatonin with a comparable drug or control drug after we justify all possible confounding factor, use the probability score approach. For example, this just shows the the preliminary data we just published last year. What we show here is we found melatonin are significantly associated about 30% reduced likelihood of SARS-CoV-2 positive rate by RT-PCR testing compared to the non-melatonin users after we justify all possible confounding factors. For example, here we tested 4 different the statistical model by justify different confounding factor we highlight here. So melatonin is supplement used for sleep disorders, and melatonin especially targeted the circadianism the pathway. So what we show here, you can see melatonin is a really magic molecule, and we also see in the COVID-19 patient transcription data, we see significant, the dysfunctional pathway in COVID-19 patient for scheduling, gene or pathway. This also highlight possibility why the melatonin can reduce the likelihood, of SARS-CoV-2 positive rate by RT-PCR testing. The second project I wanted to, share with you is after we finish the first project, we know the limitation for the drug target network is, many FDA approved drugs, they may have multiple drug or multiple target or multiple protein, but protein target, but we don't know it is. So the second project idea is, could we identify the COVID-19 drug target, just use the artificial or deep learning approach. So after we finish the project, we rapidly move this field is we call repurpose larger scale open data, use this larger scale open data just like a PubMed data for COVID-19 possible treatment identification. So we develop we call knowledge graph approach, try to leverage 24 million research article just published by PubMed. You try to build the knowledge graph, and this knowledge graph can represent the relationship between drug, protein, gene, and the disease, including the COVID-19 or other different disease as well. So this just to show our COVID-19 knowledge graph drug repositioning framework we just highlight here. So if you're interested in this work, just look at our paper, and our all code also are available in the GitHub as well. So after we get the prediction, we also use the transcription data we take from the COVID-19 patient for the bioinformatics validation as well. I just highlight a few top predicted drugs we just highlight here. For example, tauromethrin and melatonin is the top predicted drug in our network-based prediction, but we also can identify from the COVID-19 knowledge graph-based approach. But I really want to highlight one drug we call dexamethasone. So dexamethasone is a really, really top, fifth, predicted drug we have. So what we, after our paper just published 2 weeks later, the UK, published a, larger scale phase 3 trial to show that, dexamethasone can reduce the mortality rate in COVID-19 patients. And right now, dexamethasone is the only drug can reduce the COVID-19 patient, death rate right now. And, we learned a lot for COVID-19, treatment. For example, remdesivir is the first FDA-approved antivirus drug, for COVID-19 treatment. However, multiple clinical trial or phase 2 trial or larger scale trial to show that, remdesivir only have very limited, clinical benefit for COVID-19 patients. Just use the monotherapeutics approach. So, and this last project I show you is, could we use the drug combination idea to improve, the clinical benefit for COVID-19 treatment? For example, could we combine the antivirus therapeutics with the host targeting therapeutics like anti-inflammatory treatment like the melatonin we identify to improve the clinical benefit, during the COVID-19 treatment in clinical? So the idea here we use the network-based approach. We know the monotherapeutics, get very successful for multiple diseases, including the cancer treatment. We use the high dose, but this kind of monotherapy, they also have high risk for toxicity and drug resistance. Drug combination idea is we use the low dose, combine pairwise or triple or multiple drugs to treat a complex disease like the COVID-19. Our idea is in the human integral network, for example, for COVID-19, we can identify the disease module. And for the different drug, we know the drug target. Could we just look at the drug target network distance with the disease module, distance, we can better, design or predict the drug combination. So the answer is yes. So we use the strategy, or network-based approach. I just published a couple years ago. Is we call the complementary exposure-based drug combination design. We just highlight here. So what we showed, before in our original publication, for example, this so-called red circle just show disease module. We show that only 2 drug target are, complementary, synergistic way to target the disease module. This kind of, drug combination may have good, clinical benefit, but have low toxicity. We also should see interesting results for overlapping exposure. Means these 2 drugs are close to target disease module, but these 2 drugs also target each other. this kind of drug combination, they often show good efficacy in cell line or animal study, but in clinical, they have high toxicity. So using this, network-based drug combination design, we identify that melatonin and tamoxifen is a potential, treatment for COVID-19. Use our strategy. What is the tamoxifen? I think for breast cancer, most people know. Tamoxifen is a FDA-approved, the selective estrogen receptor modulator for breast cancer treatment. And based on the cell-based assay, we found that tamoxifen also have, strong or, like a micromolar, concentration across the multiple, the variants, including the SARS-CoV-2, COVID-1, and also Ebola virus. So, the postdoc well also from a lab, he also showed that use the, the competition simulation approach. She also showed that, tamoxifen may potentially block, the, spike protein interaction with the ACE interaction. This provide potential mechanism why tamoxifen breast cancer drug have antivirus, effect in the SARS-CoV-2. So based on, our, previous, computational results and EHR data and, cell-based assay, we proposed a clinical trial to test, melatonin or combination therapeutics combine melatonin with a tamoxifen, for possible COVID-19 treatment in earlier COVID-19 patients. So this project just ongoing. Just brief summary for this, three, case studies show how we try to leverage, the multi-omics data. We can, accumulate or we can collect, from by specimen from the patient, combined with the EHR data or other, the claims data we can, collect data from the clinical, use the AI approach or deep learning approach to, to identify, potential, medication for, patient treatment. So this kind of, strategy, not just for COVID. I think, Dr. Huang and Dr. Li also show, very successful cancer treatment. And now my lab also uses a similar idea for neurodegenerative disease like Alzheimer's disease, a therapeutic discovery as well. Finally, I just give, a few, discussion about this field is, so for melatonin, we also find interesting results is when we look at the different population, for example, Black Americans and white Americans in the EHR data, what we found is melatonin also showed with a reduction likelihood of SARS-CoV-2 infection rate, only we see in the Black Americans, not in white Americans. We also look at the different disease comorbidity. For example, we look at the COVID-19 patient, this patient have asthma, or this patient have diabetes, or this patient have hypertension. We see also interesting is melatonin only reduces the likelihood of SARS-CoV-2 positive rate in the diabetes patient, but we didn't see in asthma or hypertension patient after we justify all possible confounding factors. So these kind of results, really, motivate us to think about what we call the pharmacogenomic study or precision medicine for COVID treatment as well. Means each patient have different disease comorbidity, have different, clinic feature, or have different, genetic, background as well. So right now, we work together with a multiple, institute, including Broad Institute and Yale and other institute, try to do larger scale whole genome sequence study for the patient sample we collected from the Cleveland Clinical or MG, Massachusetts General Hospital or other, health care system, try to identify the genetic variation. This genetic variation, we can use it to predict the COVID-19 susceptibility, or we can predict the COVID-19 drug response, or we can identify the novel, host targeting therapeutics as well. So I just give, finally, I will really thank you, my lab member, including the Yadi and Yuan, and we'll work on this, exciting project. I want to thank you, my, institute, include, the support, include the Dr. Arizon, Dr. M, my department chair, and also, thank you very much, Laura Jehai, for, support for all the EHR, kind of collaboration from clinical. I also thank you my clinical trial perspective from the Dr. From Rena and Sujeta, and we also thank you, long, for our long collaboration in, other projects, not just for COVID, for AD project or other, cancer pharmacogenomic project as well. I also want to thank you, funding support. I just stop here, so I will go to the, with our colleague, Dr. Huang and Li for, the, panel discussion.
Oh, thank you, Dr. Chen. This is a great talk. So probably we have 3 minutes for questions for Dr. Chen. So there is a question in the chat. So it seems like you use drug target network, and that network is a population-based network. But did you consider patient-specific network, and also there are variations among different patients, like a patient-personalized network in order to identify the reversible drugs?
Yes, this is really great question, and I show we see the different drug response or a clinical across different race like Black and American or the different disease comorbidity. We are really working right now is try to leverage the clinical EHR data, large data we collected from the COVID-19 registry database, try to build the patient network and use this patient network approach for the drug repositioning study and the drug response prediction. Yes, we work on that, but we know that the challenge for this field is EHR data study is much complex than the traditional the bioinformatics approach or network approach we just present here. Hopefully we can bring some some results of preliminary discovery in the near future. Yes.
Thank you. Are there any questions for Dr. Chen? Okay, so if no questions, let's move forward to the panel discussion. So we have 20 minutes for the panel discussion. So probably feel free to type your questions in the Q&A session. And so probably while you're typing your questions here, I have some questions for the panel. And it seems to me that many of the researchers are using deep learning, like graphical neural network, those very fancy models. And one question that is typically asked is about whether you have enough sample size to train a large model. Like for deep learning models, it's very easy to get more than 100 parameters. But if we just have like 1,000, 2,000 training training samples, then how do you deal with that type of issues in deep learning?
Yeah, I think Simon also asked that question. So basically, the number of parameters will increase, dramatically. This is why I also mentioned previously 2 questions. First, whether AI model can predict the response. The second, whether we can learn mechanism synergy data. So I totally agree. Right now, we only have very small number of drug combinations, especially for single drug response, we could have much more number. But for drug combinations, I think right now we have limited number. The good thing is first is that more and more data are being generated. The second is this what I want to say is if you look at only 10 single pathways in cancer cover the many alternate alterations or dysfunction targets of many cancer patients. If we want to study save cancer patient data, discover study half of the cancer patients data, then it's possible that we only limited our study to 10 single pathways. So this is why I think in Fisher's talk, they also mentioned that that this is the reality. Many targets are not in work in one disease, but it's also, I think it's kind of polyfunction in different disease. So this is one possible thing is I presented, say, whether we can reduce number of single pathways, but focus on the complex signaling interactions among these targets. Yeah. Okay. And then we can reduce the number of parameters, definitely. Yeah. In the deep learning model, right?
Okay. So since you're talking about targets, probably a follow-up question here is about target combinations rather than drug combinations. Do you guys have an idea, or what is your opinion about identifying multiple targets or several targets working together in your work?
Yeah, but I also can quick response because Long also mentioned that's a good question. So yes, this is what I also believe that targets are the most important because it's very hard to identify a few targets from like a 20,000 genes or even more like methylations. But the key is the good thing for targeted discovery is you are not just using drug combination to train the model. For example, we can also propose to train the deep learning model using the survival of cancer patients or whether it's drug resistant or not. So basically, again, this identify the important targets could be also responsible for a drug response or drug combination prediction because they are all correlated. We call it kind of knowledge, right? Transfer. Yeah, I totally agree. Drug targets sometimes even more important. Still, I think Dr. Hong also mentioned that even right now, right? If you want to get your drug approved, you have to see, okay, what's the mechanism, what's the targets? You have to understand at that level, right?
Sure. And Dr. Huang, do you have any ideas or any other opinions?
So I guess I'm sort of approaching this as slightly different. I'm always thinking about drugs. Honestly, I am a believer we don't know exactly what our drugs are doing. That's my opinion. I mean, we know what we wanted to see, but there's this other sort of the gray area for drugs. So I think that's the beauty about letting data tell us what's happening. So with the increasing data out there, that's partly we chose the approach to, basically we have measured sensitivity. You can, I think early question from TM about, how do we apply this to other setting? I think the same principle could apply as long as you have the measured outcome. So in cancer, it's relatively easy. At least we have a low hanging fruit. We're measuring kills of cancer cells. So that's a very straightforward. To me, it doesn't matter which target you hit as long as you can achieve that ultimate goal to eliminate the target. So we have a clear measured outcome that was the response. And then we're trying to figure out, by every drug does it differently. Some we have clearly known mechanism, others are somewhat blur, but the measured outcome, it's the facts, that's our data. So our goal is to, how can we go about finding out that confirmed outcome through mining the data. Again, I think to generalize this idea, we can, as long as we can have something to measure, and that was what's the phenotypic of interest, I think we can do that. So in our case, we actually didn't use anything fancy. We used rich regression to do, to deal with multiple, we are even dealing with 1,000 samples, but we're having tens of thousand genes. So in this case, we simply just employ rich regression. We have tried other, method, basic, lasso or elastic net. Pretty much outcomes are very similar in prediction. So I see another question about reproducibility and interpretation. I very much agree with the audience about this. The interpretation about deep learning still a long way to go. Right now, we have a somewhat of a, we can create a story based on literature support. It's reasonable. That's partly how I think that's where the field is. But my hope is that we'll work out better method. Regarding reproducibility, I think that's critically important because in my mind, there's a lot of discovery that's real, but they're not clinically useful. So they are truly, the gene or the network by itself, it's truly important in projecting a drug outcome. However, the effect size or whatever you're capturing is so little that it's not prevented from being useful. So I think, so partly, I think that, the field holds computational science in the way that having a method is not it. You really have to show the reproducibility and the robustness of your work. That's why I thought Dr. Chen's work about developing the model and test it in real life in patient cohort, that's very admirable because we know these real world data could be so messy. I'm impressed that we can actually validate that finding in the patient cohort. That's probably, it's not the exact answer to your question, but that's my 2 cents.
Very nice insights. Thank you. And actually, we have two questions from the
Jack,
audience. Yes.
can I jump in for the how we can study the target combination?
Yes.
So I think my lab, we did some preliminary study how we can see the combination or synergistic way for the target combination, just protein. I would say, right now we tried the two type of strategy. One type of strategy we use is we call the human protein interaction network, because every protein or every target are in this comprehensive protein interaction network. So we can see how synergistic way this kind of protein have downstream or upstream, this kind of synergistic effect can help you to better predict how this two drug or multiple drug combination or target combination may provide a synergistic way for the drug combination design or multi-target drug combination design. This is one type of approach we call human protein interaction approach. And the second approach from the computational side, also we try, also other group try use the knowledge graph approach. So people try to take the the knowledge or the literature data we can take from the public domain to build the knowledge graph, and then we can see how the protein and the target can regulate each other from the knowledge graph, and then we also can predict the synergistic or combination for the target protein as well. So I think it's possible, and we are also try this kind of idea as well.
Probably a quick follow-up question here is how you think you will validate all the targeting and the target combinations?
Oh, this is really good question. Let me share our story for like Alzheimer's disease treatment. We also tried the target combination. Idea is, so we try to validate this target combination in the patient-derived iPS cell, neuron or microglia. We try to use the siRNA or SG RNA to knock down or knock out this the target, and then we will see the cellular response or cellular phenotype for the knockout. But I think for cancer maybe much easier, because you can just easily do this kind of functional validation in the cancer or tumor cell line. But for other diseases, it be challenging because we don't have good cell model.
Yeah, and also I think the CRISPR, even for double knockout, the proto-CRISPR is one possibility that to validate pairwise target combinations. Yeah, I think that's in 2017. Some people already proposed this idea for cancer. Yeah.
Thank you. And actually, we do have some questions from the audience. So I see someone raised a hand already. So probably Yao Liang, and you can just ask your questions, or please type your question in the Q&A session. Okay, so probably we can move forward to some questions already typed in. So Dr. Chen mentioned about the difference in Black and white African, and how would you recommend us considering such demographic difference? Or probably another question is about how do you really think such method can be personalized to each individual patient when we consider all the demographic or any other information that we can gather for each individual?
Yes. This is a really good question, but obviously we need to be careful to interpret this, Black and white American, drug response results, not just demographic or race. They also have other, big important factor we call social economic, factor as well. For example, what we learned is Black compared to white Americans, the income or other condition for Black much worse than the white Americans. For example, Black Americans have more diabetes or more other disease comorbidities. This is why we see in the follow-up study, we only see this melatonin have good response in diabetes, comorbidity patient compared to the hypertension or others. So obviously here we need to very be careful, but this is only the association study we identify for Black American and white Americans, not the causal inference, results. I think before we can apply this kind of results for clinical, we really need to do the causal inference, results before we can implement the clinical. And another strategy is we also perform the, placebo-controlled clinical trial right now. So we really wanted to test what the difference between the drug response in Black American and white Americans, because we have unique, Black American cohort we can collect it from the Metro Health, healthcare system as well. So we really wanted to test this kind of, association in the real, the clinical trial, like a placebo-controlled trial, to confirm that before we can implement or we can apply for the patient care.
Thank you. Thank you. So let's see, we have some more questions. So as for now, all the speakers were talking about like cancer. So cancer is the major disease that we are focused on. But what do you think about those deep learning methods, and also your network medicine type of network can be applied to treat other diseases like mental illness or suicide, death, or in general other diseases rather than cancer?
So first, I can just brief comment that I think this is all possible. Like I mentioned, I only show the COVID-19 study, Dr. Huang and Dr. Li show very beautiful for cancer study, and for mental disease and society death is possible. But the challenge for this kind of disease or other rare disease is we don't have omics data or genomic large scale genomic data available or clinical data available. This limited our the omics-based or multi-omics data approach for this field. If the data available for this disease, the approach we present here should be universal applied for this disease. And for suicide deaths, my colleague from the New York, they also apply the EHR data for this kind of, the drug repositioning, and the preliminary data also show that successful as well. So I just stop here and leave to Dr. Huang.
Yeah, I agree with Feijun. So basically, the methodology itself is, is kind of workable, I think, for, other disease, right? So, but depends on whether the availability. I think this is also consistent with comments is like, so even for, I want to say, there's a team's question is the interpretation of deep learning model is not clear, or reproducibility. So this is the same thing. So even for cancer right now, for like the mechanism or synergy still not so, right? This is why we are in clinical side, we are still lack of really effective drug combinations, right? Although this idea is, I think many years ago, the doctors had been testing, say, whether we can use drug combination target on, the upstream or downstream of single pathways. But as you can see, it's really hard to understand the mechanism because I think the two challenges. First, the disease single pathways are complicated. The second is it's also static status of the network. Once we perturbate the network with drugs over time, for example, acquired drug resistance, we don't know in prostate cancer. I believe Dr. Li Long also have some idea. Say once the patient treated with some medicine, the acquired drug resistance probably is totally different from the previous one. The same situation here. Once we perturbate the network with some drugs, the network will be perturbated, say some proteins expression level up or down regulated, or the connection of the network will be changed. So I think still, even for cancer, I think this is also a good, data resource. But I mean, again, we are still lack of very advanced AI. We still need to work hard to get, more models to really understand what's the mechanism of, disease and drug combinations. Yeah.
And Dr. Huang?
So my 2 cents really, it's about data. We need the data, and I don't think we are quite there yet, but I think it's nice to have this kind of conference to think about down the road what to collect. Everything come down to, of course, the bigger the number, the better, because that would allow us to address many different aspects of it. Just in the question about Black versus white, this ethnic difference, Honestly, I think the issue, I can see that partly it's in the implementation, but the other part, if our training data are mostly Caucasian or others, that would forbid us from look into the critical question about these ethnic diversity or other things. And that's exactly the case in cancer. For example, we're using these cell lines. We have looked carefully into, can we utilize them to address ethnic difference, to look for genetic underlying cause. The issue is similar to many of the clinical trial, the representation for minority is only about less than 10% in the collection of cell lines. And that's similar, representing how we look at clinical trials nowadays. So without that good representation of diversity, we won't be able to solve that problem. And sort of a selfish part, if you could, for me, study drug information, I would also say moving forward, collecting drug information is critical, because a lot of our model is projecting what would work. But when you look into real world data, oftentimes we pay a lot of attention to disease. We have all these information about the disease, the diagnosis and others, but very rarely, even though medication are constantly given, that wasn't the easiest thing to assess through the EHR or other information. To have a better framework to collect those treatment information, I think is critically important, not only for building the model, but also for down the line validating the model and apply the model. The last thing I want to say is that, everything we talk about today, mostly we haven't get to that can of worms about longitudinal information. Talking about resistance, I think for the most part, we're discussing about sort of on-site, the initial resistance to certain drugs. We, even with CRISPR and other things, we're more talking about, the target interaction is about at this snapshot of time, if you have these two things, how they can work together. But we well know drug is a big stimulus. With drug perturbation, you're changing the transcriptome, you're changing the genome. So I don't think the work presented, we're not quite, again, due to the limitation of data, a lot of those develop resistance is a major problem for cancer and many other disease. And that's not, I think it's the work we've discussed today, most of them are not focusing on that. So that's something I believe it will come. But again, mostly I just want to advocate to collect more data, more diverse data, try to collect drug information and longitudinal data that will enable us to do more work.
Thank you, Doctor, for very nice insights. So we have probably just 1 minute left for this session. And the last question is for every panelist here, for all the 3 speakers. So if you can think of just one thing about AI in digital health for drug repositioning, either about the data or the AI technology, what is the most challenging and the most exciting thing that for you? I mean, just one thing for you. I mean, if you want to spend the next 10 years on one particular thing on AI, what is that?
I just go very quick, from our research experience is I think we believe, AI, technology, provide a very powerful tool for drug repositioning and therapeutic discovery, for challenging disease. But, the major challenge for us right now is the communication, between the different, field, right? From the basic research field, for computational field, also from the, pharmacology and chemistry from, the company as well. So also from the government, like a regulatory side, as well. How we can, better sell our AI model or sell our, larger scale or hundreds of, prediction to narrow down to get the final, the candidate for cell-based animal or preclinical validation. So I think we really needed to communicate each other across the multidisciplinary. What I would see is in the next 10 years, we will see more and more multidisciplinary research, not just our computational sites work alone. This is what I see in the next 10 years, and make the drug possible or get us successful for this field.
Yeah, multidisciplinary research and integration and implementation. Yes. Thank you. And how about Dr. Li?
Yeah, I agree with Fei-Chun. So I think, also Dr. Huang mentioned without data, our understanding is at a static level and with very small level. we know many things, right? Methylation, microRNA, gene expression, proteomics data, but I don't think there's a model can integrate this data together. Because of what? I think two things. One is we don't have a label to learn, right? What should we learn? And for example, we learn drug combination, but we don't have any effective drug combinations. How can we learn that, right? The second is, so come to the question is, Fisher mentioned is, yes, we have to integrate the domain knowledge, like different disease experts definitely have much better understanding than us, than pure computation guys, and also more experimental validation can help us. So this is come to the technical challenges of AI itself is how can we make really intelligent AI models without so many parameters. I think this is a challenge, right? Right now it's pure data driven. It's good and we can train the parameter. It's bad. It's not real intelligence. We are just identify the associations rather than causal. Yeah.
Yeah, so it's about interpretability of deep learning models and more intelligence within the deep learning models. Thank you. And how about Dr. Fang?
I guess I am an optimist. I really love the fact that we're applying these, even though we're early in the AI development, to take advantage of data, apply for drug repurposing. I know there's some doubt about whether things has been approved yet. I think given the time, we're making huge progress. We're just not quite there yet. I am a believer that we can take advantage of existing data and make speed up things, both for single drug development, biomarker development, and combination. So I believe there will be, I believe that's also the case, not only academic, but also industry. Everybody is thinking about that. And there's limited number of resources to be made into drugs. So I am a strong believer, employ computational science in drug development. It's a critical part of this for human health. Yeah, that's my belief. I saw like TM again make a comment about links data is mostly in cancer cell lines. So his question about applicability in other disease system. I would say I'm with you. Initially, I thought I'm lucky to study this in cancer. But honestly, in our recent practice, we have tried to use the model we built in cancer and apply it to, for example, TCGA, there's normal tissues and other things. Surprisingly, I guess cancer is derived from normal, so we do see some similarity there. So I guess I have this slightly change of mind. I still believe cancer is the direct application parts, but a lot of these tools currently used for cancer, I think there's a clever way to use them for other disease too.
So it's about the generalizability of current models towards other diseases, right?
Yep.
Oh, thank you. Thank you again for the three speakers. It's very nice presentations and also very nice discussion and insights. Really appreciate that. And I think that will conclude our session, and we will have 20 minutes, until 10:10 for a short break. And please come back for the next section on imaging and AI for biomedical imaging. Thank you again for the speakers, and thank you for the audience for attending this session. And I'll see you back in 20 minutes. Thank you.
Thanks, Chair.
Okay, bye.
Thank you. Bye.